Okay, so before we start with photosynthesis, I want to go over chloroplast structure. So basically the anatomy, microanatomy of the chloroplast. And these are used in David Knuffke's Prezi notes. They go along with the Campbell and Reese AP biology textbook. So first off, the chloroplasts are very similar in structure to the mitochondria because they both started off eons ago as single-celled organisms, a single-celled, basically cyanobacteria that got engulfed through endosymbiosis. So that it has that same double membrane structure as the mitochondria. It has outer and inner membrane, where the outer membrane was originally the vesicle that the cell that ate it put around it, and the inner membrane is its original prokaryotic cell membrane. Now, just like the mitochondria, there's some specialized vocabulary. So the cytoplasm that is inside the chloroplast is called the stroma. I apologize for biologists' poor naming. It sounds very similar to stoma, which is a hole in the underside of a leaf to take in carbon dioxide. So the spelling does matter on this one. So stroma is basically the cytoplasm. It's the equivalent to the matrix in the mitochondria. So the stroma has all the enzymes, it has plasmids, it has ribosomes, and it has DNA. And the stroma is where the light independent reactions take place. So that's where sugars are made, where the Calvin cycle occurs. The thylakoids are these sacs that are kind of stacked up on one another. So an individual one is called a thylakoid. Many are thylakoids, and the whole stack is called a grana. All of the stacks. One grana is plural, one is called granum. So if you see it either way, it's the same thing. And you can see they're kind of cut apart there. This is much more intricate than the mitochondria is. So the mitochondria does have its inner membrane folded into cristae. The thylakoids are analogous to that, but there's way more of them. And you can see in the cutout here, you know, how they zigzag back and forth. Each granum is connected to another one, okay? It's a one continuous membrane that's just super invaginated to really, really maximize surface area, because this is where the light dependent reactions take place. So every little bit of that membrane is packed full of photosystems and ATP synthase and all of the other proteins and enzymes that are needed to capture light and convert it into chemical energy. So it's super maximized. Sometimes people describe these as looking like stacks of pennies. I think the whole thing looks kind of like a Mentos with little stacks of pennies inside. Here's what it looks like in a a transmitting electron microscope, a TEM. Very small. This is 1 micrometer. So this is 1 meter times 10 to the negative 9. Very tiny. Okay. And the chloroplast, it does photosynthesis. So photo means light, synthesis is making. Okay, so we're using light to make sugars. Oops. Back here. Okay. And the end product, one of the end products of photosynthesis is oxygen. Oxygen is a waste product in this chemical reaction. And in fact, when cyanobacteria and plants first evolved, they actually caused a massive extinction event due to the rapid increase of oxygen into the atmosphere. Oxygen is actually fairly toxic, highly combustible. If you remember from chemistry about combustion reactions, they always have oxygen in them. We called it the Great Dying. Many not just the individual organisms, but species and genera went extinct due to this. And it took a while for aerobic respiration to evolve. So prior to this, we did have anaerobic respiration, the glycolysis. That's one of our most ancient metabolic pathways. Everything, every cell uses glycolysis, but not all do aerobic respiration. And aerobic respiration evolved in response to the selection pressure of massive amounts of oxygen in the atmosphere. So way back when, the oxygen was much higher than the current roughly 21% that it is now. Okay, chloroplasts are green. There are other plastids that are different colors, but chloroplasts are green. And it's because they use chlorophyll. There's two kinds. There's chlorophyll A and chlorophyll B, and they reflect green light. The green light isn't used by chlorophyll. Chlorophyll uses all the other colors of light in the spectrum. Now, out of the classification system, we have 3 domains. So we have bacteria, archaea, and eukarya. Okay. Some bacteria can do photosynthesis, but they're prokaryotic cells. They do it in a different way. So we're going to kind of ignore that. It is important, but it's done in a different manner. So we're going to focus only on eukarya, and out of eukarya, only plant cells have chloroplasts. You may have heard in the news recently, oh, we've discovered this sea slug that does photosynthesis, or coral does photosynthesis. Well, in reality, those are animals, but they have a symbiotic relationship with the algae that lives with them, and it's the algae, which is a plant, that does the photosynthesis, not the animal part. Okay, it's the same with lichen as well. A lichen is part algae and part fungus. The fungus does not do photosynthesis, the algae does. So these are from a duckweed leaf, which is an aquatic plant and pretty common. You can see it's a plant cell because it's mostly rectangular. And then these are the chloroplasts. Here, I think they kind of look like squished up onions that are green. And this is colored. It doesn't look like this under the microscope. These will look green, but the rest of this won't look that way. They're dyed. Okay. All right. So as far as energy processing goes, like I said, the mitochondria and the chloroplasts are very similar. And you could get asked on the exam to compare the two or contrast the two. We study them together too because they're both ways of transforming energy. Remember the laws of thermodynamics, matter and energy are neither created or destroyed in this universe. They simply transform from one version to the other. So the mitochondria is transforming the wavelength energy of light into chemical energy, or excuse me, the chloroplast is doing that. And then the mitochondria is converting chemical energy into other forms of chemical energy. Both of them make ATP. They do it in a different way, but they both make ATP. They both have ATP synthase to do it. They both have double membranes, the outer and the inner, and they both have an intermembrane space. They both use that intermembrane space to accumulate a high concentration of hydrogen ions and then power their ATP synthase by funneling those hydrogen ions through that enzyme. They're both what he calls semi-autonomous organelles. So they reproduce on their own independent of the larger cell, have their own DNA, they make their own proteins, they have their own ribosomes. And how this is explained is, once again, going back to the endosymbiotic theory. So we know that the mitochondria came first based on fossil and molecular evidence. So a larger cell engulfed the mitochondria, it became a part of it. For some reason, it didn't get digested, and they have co-evolved together for roughly 3 billion years. Okay. Mitochondria are not the same as those ancestors. They've lost a lot of genes along the way, and now they're obligated to live inside a eukaryotic cell, just like chloroplasts are. They cannot survive on their own outside of the eukaryotic cell. The mitochondria, like we did in one of our practices, they've lost the genes to break down glucose. They cannot break down glucose into pyruvate. Then we know at least one more time another symbiosis event occurred with chloroplasts. So the ancestor of chloroplast again got engulfed by a cell that was the ancestor of all the plant cells, called endosymbiosis. Okay, but there is more. Within plants, this actually occurred more than once. Chloroplasts are just one type of plastid. There are more. You only need to know chloroplasts for the exam, but it doesn't hurt you to be aware of the others. And we can track these. We can track their evolution through genomic analysis because all of these plastids have their own DNA. So we can see exactly which ones came first. But here's the proposed lineage. Like I said, you don't have to know these, just know that they exist. Okay, so this one, an amyloplast, is for starch storage because the name of starch is amylose. Proteinoplast stores protein. So those ones at least make sense, but there are some other ones too. And then we can see this in different algae as well, track their genetic lineage of endosymbiosis. There's actually a lot of debate now amongst taxonomists, those who classify life, and I'm pretty sure within my lifetime, rather than having 3 domains and then 4 kingdoms within domain Eukarya, I'm pretty sure the protist kingdom is going to be reclassified because the only reason they are there together is that they're single-celled. But there are single-celled algae that are plant-like, and there's other single-celled organisms that are animal-like, and others that are fungi-like. So the more we learn through genomic analysis, the better our microscopy gets, the more we're seeing that the way we've organized our evolutionary relationships is not done properly. And there's even a lot of debate about reorganizing the domains and not even having Eukarya as a domain because if we go back to this, the nucleus was actually an endosymbiosis event from an archaea. So we've got, if you're a plant, an archaea and 2 little bacteria that are all working together inside a larger cell. So we're actually, it's more properly called, instead of a tree of life, we call it a braided ladder now because there's so much what we call horizontal gene transfer and so much symbiosis going on. Okay. So now we're going to talk more in depth about photosynthesis. And technically, from an ecological standpoint, those organisms who practice photosynthesis are properly called photoautotrophs. Photo is light, auto is self, and then trophy is its feeding method. So there are also heterotrophs, like we are. We are heterotrophs because we practice other feeding. We eat other organisms to stay alive. And even if you're a vegetarian, you're still eating other organisms to stay alive. You eat an apple, that apple is alive while you're eating it, unless you've cooked it and you do an apple pie. So. Please understand that plants also have mitochondria. Okay? They use chloroplasts to make their sugars, but then they need their mitochondria to be able to turn those sugars later on as needed into ATP. It's a reciprocal relationship. Photosynthesis, sugar, mitochondria breaks down sugar, ATP is being made and used up through that whole cycle. So these are the equations. And if you look at it closely, one is simply the opposite of the other. This is actually an equilibrium equation when you think about the entire Earth as the system that this equilibria works in. You should know and be able to write and recognize this equation. Don't let it scare you. Okay, if balancing equations isn't your thing, this is a simple one, but 6 carbon dioxide plus 6 water, so these are the things the plants eat, will yield 1 glucose, C6H12O6, plus 6 oxygen molecules. So you should, this part should be easy. You just have to remember that oxygen does not exist in the atmosphere as just one oxygen. The oxygen gas is O2. And then this is just the chemical formula for a monomer, okay, hexose sugar monomer. It's always going to be 6 carbons, and the way the formula works is the oxygens and the carbons are equal, and the hydrogens are always double. And aerobic cell respiration is just the reverse. Remember, plants are not the only photoautotrophs, so other organisms do photosynthesize, and a lot of them are protists or bacteria, and it's the cyanobacteria that are the ones that do the photosynthesis. Now, the protists, like I said, we're probably going to wind up reclassifying them as soon as science reaches a consensus on that, because the protists, there's much growing evidence that they really are, their ancestors were a common ancestor with modern plants. And the cyanobacteria, there's a lot of genetic evidence that they were the ancestors. They shared a common ancestor with modern chloroplasts. And yes, most of the oxygen that's currently being generated in our atmosphere actually comes from these single-celled organisms, prokaryotes and eukaryotes, that live in oceans. So on the land, the terrestrial plants do play a factor, but not nearly as large a one as the oceans do. Photosynthesis occurs in the green parts of the plant, so the leaves. Plants do have other organs. They have roots under the ground. Some of them have special storage organs, like a tuber, a potato, or a bulb, like an onion, where they store up sugars for later usage. But those parts under the ground do not do photosynthesis. There's no light going under the ground, so they don't need to do photosynthesis. It's just the leaves. The leaf has its own special anatomy where it has different tissue types within it, and one of the top layers of a leaf is full of mesophyll cells. It's called the mesophyll layer. Those mesophyll cells are cram-packed full of photosynthesis, or full of chloroplasts for photosynthesis. Okay, so just like we talked about with the mitochondria, if you want to get in shape, you go to conditioning and your muscle cells make more mitochondria and you have a higher baseline. Same with the plants. Plants that are exposed to more light more often, they get conditioned and they make more chloroplasts to take advantage of the light. All right, so light. The visible light that we see is actually a very small part of the entire electromagnetic spectrum. Okay? Electromagnetic radiation goes all the way from gamma rays to radio waves. So gamma rays are really high energy, and as you move towards radio waves, those are really low energy. So the wavelength of light determines how much energy it has. And it's represented by lambda usually is what wavelength is. And they're listed up there. So everything to the right of visible light, the wavelengths are long enough and the energy is low enough that that radiant energy actually does not interact with physical matter. So it won't mess up our DNA and cause mutations. So, and this has been studied, okay. Radio waves do not cause cancer. You know, your cell phone's not going to give you cancer. It just passes right through you. But on the other end of the spectrum, at wavelengths with higher energy, shorter wavelengths, those will interact with physical matter, and they can break down covalent bonds. So they can cause DNA strands to break. And we do have cellular machinery to repair it, but a lot of times there's mistakes, especially over time if you keep having that radiation. And mutations can cause cancer. So we're going to talk about that when we come back from break. And you can see this with UV light. UV light can cause you a sunburn. The energy from the light can actually physically damage your skin, which is why we need to wear sunscreen, especially here in Florida. Other animals can see in UV or infrared, but not us. We can only see the visible light in the middle because we have special nerve cells that are called photoreceptors. They receive light that can, and action potential can be triggered by that light, and we actually see it as color, the colors of the rainbow. You can actually see the colors of the rainbow if you use a prism to split white light. The prism actually slows and sorts the wavelengths, sorts them based on how long they are, and so you get that rainbow effect. You can also see natural rainbows. How the light's being split is through water droplets in the sky. Now, plants don't have photoreceptors the way we do. They don't see, per se, they don't have a brain, but they do use the light of the visible light on this electromagnetic spectrum. Now, plants can also get cancer, and they can also be damaged by UV rays as well. And they have natural protections, just like various animals do, like having darkened skin. But for a plant to get a cancer, it's not as bad as it is for a human, because plant circulatory system doesn't move as much as ours does. So when they get a cancer, it tends to stay put. And so if you've ever seen a big like bubble thing on the side of a tree, call it a gall, but that's a tumor, a cancer cell. All right. So chlorophyll is found within the chloroplast, and it's chlorophyll that actually captures the light energy. So chlorophyll, what it is chemically, is a pigment. So it's actually a protein, okay, but it's a special class of protein that we call a pigment. If you've had any art classes, you've probably used that term before. Pigments make colors. There's different ways to make colors because you can make a color with a prism out of light, but a physical molecule that makes a color is called a pigment. Chlorophyll is one such pigment, and plants have two main varieties. There's chlorophyll A and chlorophyll B. So this is the main photosynthetic pigment, but it's not the only one. There's other ones like carotenoids. They're kind of yellowy orangey, and also xanthophylls, which are bluish purplish. So you can think of a carotenoid pigment as like the main pigment in carrots, or xanthophylls like blueberries. Okay. They're called accessory pigments. So the way that color works in a pigment is the color that you see with your eye is the color that's reflected. So for chlorophyll, they reflect green wavelengths of light, and those wavelengths bounce off chlorophyll, hit our eye, and then that stimulates our photoreceptors, assuming we're not colorblind. But the chlorophyll pigment actually absorbs all the other visible wavelengths. So they absorb the red and the orange and the yellow and the blue and the violet. Okay, they absorb those, and the energy from what they absorbed is what's used to drive and power photosynthesis. For carotenoids, they reflect the orange-red end of the spectrum, but they absorb green and blue and violet. And then xanthophylls, they reflect blue, but they absorb all the other colors. So that's how that works. So technically, the color that you perceive is not actually the inherent color of an object. It's the color that is reflected away from the object. But the accessory pigments are there to help absorb that green wavelength of light so that the plant isn't wasting any of the light energy that's coming in. It's more efficient that way with the accessory pigments. And you can actually see the accessory pigments in places that are not Florida, that have winter, when the leaves change in the fall. So the plant will actually digest the chlorophyll pigment in its leaves through a process called abscissence. And it breaks, it's a protein, so it hydrolyzes all the peptide bonds and reabsorbs those amino acids and uses them later for other cellular processes. So it's not being wasted. That process starts first, so you can see once the chlorophyll, the green is going away, you can see the underlying accessory pigments, and then that's why you have like purple or orange or whatever leaves in the fall. I got ahead of myself. You can also see some of those accessory pigments in some plants that will change their color in response to light levels. So there is a plant called Coleus. We use it in the lab a lot here. But Coleus, if you put it in full bright sunlight and let it grow for long enough, all its new leaves are going to be green. But if you grow it in the shade, say under an oak tree, its new leaves will be purple or gold. It is really, really quite pretty plant too. But so plants have, methods where they can respond to environmental conditions. Now, scroll up a little since I already said this. So on the right here, this one is an absorption spectra. Spectra is plural for spectrum. So you can take with scientific instruments and measure how much of each wavelength, what quantity of wavelength is being absorbed. Okay, and then it makes these graphs. So you can see chlorophyll A is here. So it has peaks over here in the blue and the violet, and it has peaks over here in the orange and the red, but it's very low in the green. Okay, so it's not absorbing green. And the same with the chlorophyll B. Chlorophyll B is shifted a little bit over, but no green. Versus the carotenoid, it reflects all of this. So there's none here, but it's really good at absorbing this green and blue and indigo light. The action spectrum is which of these wavelengths are used to do photosynthesis. So these are all the possibilities of wavelengths that a pigment can absorb. These are all the possibilities that they can use. So it doesn't matter if you absorb it, if you can't use it. There's very little going on here in this like 570 range. So none of these pigments really absorb it. They've evolved not to because it wastes energy. Now, we in the lab can measure this with a spectrophotometer, which is not shown in this picture. This is like the older way to do it. So we just have a little machine. We put your solution in the machine, close the lid. It shines light through at different wavelengths. The light is split by a prism. And then we measure what comes out on the other side, and then the machine will make this for you. It's actually pretty cool. So you can actually extract different pigments from these plants and see this for yourself. This is something we typically do as a wet lab in this course, but due to COVID, we were unable to. So I think I have a gizmo that we can do potentially. So moving on to the biochemistry. We can skip over that. We can skip over that. So there's two main parts to photosynthesis. This is easier than when we did cell respiration, because cell respiration had glycolysis, link reaction, Krebs, and then the ETC. So there was 4. This one only has 2. There's different ways of calling them. Sometimes the old way was to call it the light and the dark reactions, but it's a little misleading, because the dark reactions, they can happen in the light, they just don't need light. So we've changed that, but if you read an old paper, you might see that. That's what they're referring to. Now we like to call them the light-dependent reactions. The reaction can't proceed without light because it's dependent upon the light. Or sometimes we just say light reactions. And then the other reactions we call light independent. They don't need light to occur. This is also called the Calvin cycle. In the light reactions, ATP is made. So light is gathered in the chloroplast. It uses photosystems and electron transport chains to move hydrogen ions and then make ATP. That part happens first, and the hydrogen ions are obtained by using the light energy to split water. You split the water into its hydrogen and its oxygen, and the oxygen is what is the waste product. But in the Calvin cycle, the ATP that was made prior and the NADPH, which is a high energy electron carrier, just like NADH, those are then used to make sugar, to convert carbon dioxide from the air into a sugar. This is called carbon fixation. The products of the light reactions are the reactants in the Calvin cycle and the light independent reactions. So if you don't have step 1 going on, step 2 cannot occur. And it may be a helpful mnemonic for you if you get confused between the NADH in cell respiration and the NADPH in photosynthesis. Well, the one with the P goes with photosynthesis. Okay. So the chlorophyll, the light shines on the chlorophyll. And what happens at a chemical level is that the light is absorbed by electrons, and that causes them to jump energy levels. They become excited. In chemistry, you probably learned about like the S level and the P level and D and F and all that. Like that's what we're talking about. So photons of light, which is when light is acting as matter, a photon happens in a specific like quantity, and that photon imparts its energy on the electron, and the electron jumps up the energy level. So it's moving faster, has more energy. Because it's moving faster and it has more energy, it can break off the chlorophyll molecule, and then it moves down the electron transport chain. We say that the electron has become excited. Now, there are two photosystems, which is a big protein complex that's embedded in the membrane of the thylakoid. There's protein, there's pigments, they're all together, okay? And then those photons of light get directed into the reaction center where they excite the electrons of these molecules. The electron comes off and then it moves down the electron transport chain, so through the inner part of the plasma membrane. And there's two photosystems. Okay, wait. There we go. There's photosystem 2 and a photosystem 1. And we're bad at naming things in biology, so photosystem 2 happens first. Photosystem 1 happens second. Okay. As far as like where they're located. And you can see in the bottom picture here, that's the ribbon model, like we looked at earlier with proteins. It's the ribbon model of how complex this photosystem is. And they each function at slightly different wavelengths of light. Now, the chlorophyll, once it's lost its electron, it has a lack. It needs to replace that electron to be chemically stable. So it gets a replacement electron through water, okay? Specifically the splitting of water, because it cannot keep, it only has a finite amount of electrons. It can't keep sending electrons down the ETC without replacement. So the water will go through a process called photolysis. Photo for light, lysis for splitting, just like we talked about hydrolysis. In hydrolysis, you use water to do the splitting, but in photolysis, you use light to do the splitting. So for every for every 4 protons and 1 molecule of oxygen gas, 2 waters are consumed. Okay, so remember, a proton is a hydrogen ion has 1 proton and 1 electron. So we strip the electrons off, send those down the ETC. What's left is the hydrogen ion, which is also called a proton. The oxygen gas is a byproduct. It gets released back into the atmosphere, and then it is used for cellular respiration. Then what happens is the protons, the hydrogen ions, actually get pumped into the intermembrane space, or the thylakoid space, so in between. And those hydrogen ions obtain a really high concentration. So a concentration gradient is built up in this thylakoid space, and they will then diffuse through chemiosmosis through ATP synthase, and this ATP synthase works the same way as it does in the mitochondria. The ATP synthase turns as the hydrogen ions flow through it, and that turning provides the energy to make a bond between ADP and an inorganic phosphorus. Okay. Now, we don't call this oxphos like we did with cell respiration, because technically the energy for making ATP in cell respiration is coming from glucose oxidation. So that's where the oxidative comes from, and the phosphorylation is for the ATP. Instead, we call this photophosphorylation because it's the light, the photo, that added the phosphorus to the ATP. Okay, so here it is kind of zoomed in. You have photosystem 2 happening first. We have a cytochrome complex. You don't have to know all the names of these things, okay, but you do need to know photosystem 1. But this is the ETC. The electrons are moving through this chain, and they are what's powering the protons, hydrogen ions, to be pumped into here. The NADPH is made down here, and that moves on to the Calvin cycle. And here's the water, where the water is being split. All right. So the electrons are traveling down the ETC, but kind of at the end of it, they do get transferred to this enzyme. You don't have to know the name of it, but this enzyme will reduce NADP plus to become NADPH. So the other product of this reaction is NADPH in addition to the ATP. The NADPH will carry those hydrogens onto the Calvin cycle. That was happening in photosystem 2. Photosystem 1, it's cyclic. They get kind of recycled, and that makes ATP. The cyclic versus the non-cyclic, that was focused on more in the old exam, so don't worry about that for this exam, but it's still here on the PowerPoint. Okay, and then the Calvin cycle requires 9 ATP, 6 NADPH for every sugar produced. Now for this one, Calvin cycle is really complicated when you get into all the nitty-gritty that they're not going to have you do. So on this one, you don't have to memorize like the numbers like you did in the last chapter. Over that. All right. Then we move to the Calvin cycle. So the Calvin cycle must have carbon dioxide to power it. If there is no carbon dioxide, or if there's reduced carbon dioxide, the Calvin cycle, it will either not function at all, or it's going to slow down. Just like you can't, bake cookies if you're out of eggs. Don't have carbon dioxide, you can't fix that carbon, you can't build a sugar because you don't have a building block. So there are 3 phases. First one, here we go. The first one is called carbon fixation. So fixation is when you turn an inorganic carbon into an organic carbon molecule. So carbon dioxide is considered inorganic, but glucose is organic. So this happens through use of an enzyme called rubisco. Its full name is there, ribulose bisphosphate carboxylase. All that means, ACE, it's an enzyme. Carboxyl means it adds carbons. Bisphosphate, there's 2 phosphates on the ribulose. Okay, so it's just a sugar. So it's an enzyme that transfers carbon dioxide onto this sugar that's already there. It is a cycle. We talk about this as the starting point of the Calvin cycle because that's where carbon dioxide enters. So the carbon will get added to the existing molecule that was already there. Then, next step, that molecule becomes reduced, and ATP and NADPH are used to do the reduction. Okay. RUBP is the name of this molecule. You don't have to know the chemistry or these names. Okay. The next step is called regeneration, is where ATP is used to reconstitute the RUBP from the G3P. Again, you don't have to know all those names. Just know that it's a cycle. There's a lot of chemical rearrangement going on here. It actually takes 6 turns of the cycle to complete a full molecule of glucose because there's only 1 carbon coming in at a time from the carbon dioxide, but glucose has 6 molecules. Okay. So you have to make 6 G3Ps. Go around the cycle 6 times. Okay. for you to make one sugar. And then from there, outside of this cycle, the plant can use that sugar for cell respiration and make ATP again, or it can chain it up with other sugars and make disaccharides or polysaccharides, depending on what's needed. Okay. Go. If there's a name smacked on something that you learn about in science, it probably means somebody with that name discovered it. So Calvin. Okay. Now, the way evolution works is it takes what you have and it modifies it to use for whatever the situation at hand calls for. There's no intent. If there were intent, we humans wouldn't, have weird little quirks like getting the hiccups, which is a remnant from when our fish ancestors used their diaphragms to suck in water. It's still there. There are other, bad evolutionary quirks like our knees and our lower backs, because those evolved in quadrupeds first. But then we decided to start walking upright, and evolution didn't account for that. So we have knee and back problems. Same thing here. So Rubisco, the enzyme, evolved first before there were high concentrations of oxygen. Because remember, oxygen was a byproduct. That's what caused the Great Dying. So it turns out that the chemical environment that Rubisco is in now is not the environment that it originally evolved in. So it just happens to work good enough that nothing else has been able to outcompete it, because it was already pretty widespread before we had the high oxygen gas problem. So it's an enzyme. Its active site actually has an affinity for oxygen, which is not good for photosynthesis. We want to get the oxygen out of there. This causes a problem. So on occasion, instead of using carbon dioxide, you get oxygen oxygen entering the Calvin cycle, which is wasteful. It's very wasteful because it just stops the cycle. It uses the ATP up, doesn't make any sugar, but we still get just enough sugar that plants still have a competitive advantage. They've survived to reproduce and pass their genes on, so that's what we have. It just is. It's an evolutionary byproduct. However, if the plant keeps its stoma open, or stomates, which is the holes in its leaves, as long as those are open and gas is being exchanged and we get rid of the oxygen from the leaf and CO2 comes in, no real problem. The plants don't always want to keep their stomates open, because the stomates also lose water. And you do want a little bit of water going through for the process of transpiration. That's the process that pulls water from the roots up to the leaves. But if it's really hot, really humid, like in a desert, or not humid, sorry, in a desert where it's really hot, you don't want that. You want to minimize water loss. So other plants have evolved slightly different pathways, alternatives to the Calvin cycle, to minimize this photorespiration. So they're not losing water and they're not wasting ATP. So there are different names for these. There's C3, C4, and CAM. So the C3 is what we just learned about. C3 for 3 carbon. This doesn't take into account photorespiration. Both the light-dependent and the light-independent reactions take place at the same times. Oxygen and carbon are exchanged at the same times through the stomates. Sugars get transported from the leaf throughout the plant. But if you have a desert, the plant will dry out. It will desiccate is what that's called. It will dehydrate. So that's not good. So plants would prefer to keep their stomates closed, but with a closed stomate, that will eventually lead to a buildup of oxygen within the leaf and a reduction of carbon dioxide within the leaf, and that does not help the plant make sugar. So other plants have evolved adaptations around that. So C4 plants, this would be a plant like corn. Corn is a C4 plant. In this case, the carbon fixation occurs in mesophyll cells, and it uses a different pathway rather than the Calvin cycle. So they use a 4-carbon base. You don't have to know the chemistry of this. Just know that C4 uses a different metabolic pathway. But they use a 4-carbon base for this cycle, and then so it starts in the mesophyll cells, and then they transport that 4-carbon to a different cell, bundle sheath cell, where 1 carbon is cleaved or cut from the 4 carbons it was attached to. And then basically what that does is it spatially removes this cycle from where the oxygen concentrations are high. Okay, so there's like an extra step, and then it finishes out a modified Calvin cycle. There you can kind of see, so there's 2 different cell types where this is occurring. So we have an intermediate step. This is much more efficient. I know corn isn't a desert plant or anything, but it is a modification, and scientists are kind of looking into this, you know, as far as like making our crops more efficient. Okay. Now, CAM plants are different. CAM. I forgot what that stands for now. Crusulian acid something. But these plants actually separate the cycle from the oxygen through time. So they actually do their Calvin cycle in the evening, so they can keep their stomates open to exchange gases. But because temperatures are lower at night, there's less chance of dehydrating or desiccating. But what that means is they have to convert that carbon dioxide and store it up for use during the day later. And here's an example. So sugarcane and pineapple, these are CAM plants. Skip over that.